You know that feeling, right? You're minding your own business, and then a sneezing fit hits you out of nowhere. Well, it turns out there's a wild microscopic drama playing out inside your body, and it all comes down to one molecule with a serious identity crisis. And this isn't some rare thing. Let's just get a sense of the scale here. According to research from Nature Reviews Immunology, about 1 in every 3 people now has some kind of allergy. That is a massive number of us all dealing with the exact same haywire biological response. So that leads to the big question. What is actually going on in there? Who is this tiny culprit that turns a beautiful day into a total nightmare of sneezing and itching? Well, the answer lies with a very specific and very special antibody. And here it is. Let's meet the main character in this whole story, immunoglobulin E, or as we'll call it, IgE. This little molecule is the star of the show, but as you're about to see, it's kind of like a hero that's gone rogue. See, IgE wasn't designed to make our lives miserable. Its original job was super important. It was our first line of defense against things like parasites, a real biological security system. The problem is, in our super clean, modern world, this system has gotten a little jumpy. It started mistaking totally harmless things, like pollen, as deadly threats, and it triggers this massive, all-out defensive war for no good reason. So what is it about IgE that makes it so dramatic, so prone to overreacting? Well, a huge piece of the puzzle is its shape. It is just not built like your average antibody. It's peculiar. Okay, so most antibodies you might think of, like IgG, have this classic, pretty flexible Y shape. It's got a hinge in the middle that lets it move around. But IgE, nah. It does things differently. It has this extra rigid pair of building blocks right where that flexible hinge is supposed to be. And that one little change has a huge effect on its overall structure. And here you can see the result. Instead of that open, flexible YIG, EG is seriously bent, almost folded back on itself. And trust me, this is not just some minor little detail. This bent, compact shape is the key to its aggressive function in an allergic reaction. It completely changes the game. But here's the thing. IgE is not a solo act. It's the leader of a network, and it has some key accomplices that help it do its dirty work. And those accomplices are receptors that live on the surface of our cells. Let's meet the two big ones. So we've got EFKA epsilon R1 and CD23. The way to think about it is this. EFKA epsilon Ri is the hair trigger alarm. It has a super high affinity for IgE, which means it grabs on and holds on tight. It's ready to go off at the slightest touch. CD23, on the other hand, is more like the network manager. It has a low affinity, and its job is much more about regulating the whole system. Totally different roles. Now, to really understand that dramatic instant allergic reaction, the sneezes, the hives, we have to zoom in on that hair trigger alarm. The interaction between IgE and the FcFsIR receptor, that's what lights the fuse. All right, now we get to the good part, the climax of the story. We've got our molecule, IgE, and its high-strung receptor, Fc epsilon R. All the pieces are on the board, ready for a really fast, really powerful chain reaction. So let's watch how it all goes down. Okay, here is how the dominoes fall. First, IgE molecules float around and dock onto those Fc epsilon I receptors on immune cells called mast cells. You can think of mast cells as tiny, tiny grenades, and this step is basically arming them. Next, the allergen shows up. Let's say it's a grain of pollen. It comes in and latches onto 2 of these armed IgE antibodies at the same time. This is the trigger. This cross-linking yanks the receptors together, which sends a frantic go signal into the mast cell. And the final step, the grenade goes off. The cell degranulates, which means it just dumps a flood of chemicals everywhere, including the most famous one, histamine. And that chemical flood, especially the histamine, causes what we call the early phase reaction. This is the stuff you feel almost instantly, literally within minutes of exposure. The sneezing, the itching, that red flare on your skin, that is the direct result of those mast cells exploding their chemical cargo. But it's not over yet. Other chemicals that were released in that first blast start to send out signals for backup. This kicks off a second wave, the late phase reaction. This happens hours later, and it calls in even more inflammatory cells to the area. This is what's behind that lingering, drawn-out inflammation that makes you feel miserable long after the pollen is gone. So now that we've met the culprit and we've seen the chaos it causes, The obvious next question is, how do we stop it? Well, this deep understanding of what's happening at the molecular level is exactly what is leading to brand new, targeted treatments. I mean, it's pretty logical, right? If you want to stop a crazy chain reaction, the smartest thing to do is to break the very first link. The goal here is to stop our main culprit, IgE, before it ever gets a chance to arm those little grenade cells in the first place. And this is not just a theory. This is really happening. As the source material points out, therapies that go directly after IgE have been a huge success. The prime example is a therapeutic antibody called omalizumab. So how does omalizumab work? It basically acts like a molecular bodyguard. It's a custom-designed antibody that hunts down and grabs on to any free-floating IgE in your system. By latching on, it physically blocks the IgE from ever docking onto those hair trigger receptors on your mast cells. And it's simple. If you can't arm the mast cell, the allergen has nothing to trigger. The whole allergic chain reaction is stopped dead in its tracks before it can even get started. This whole approach has been a massive game changer, and it really leaves you with a fascinating question to chew on. We've focused on stopping IgE, that first link in the chain, but we also learned about the receptors, the signals, the whole network. So the question for the future is, what other weak points in this complex system can science target next to finally bring some relief to that 1 person in 3?